Hello, my dear audience, and welcome to another episode of Let's Talk Micro. I am so happy that you are here with me. If this is the first time that you are listening to Let's Talk Micro, well, thank you for choosing it as your podcast or one of your podcasts. I appreciate it. And as I am recording this, I just got back from ASM Microbe 2025 in Los Angeles. What a great experience, and I'll be doing an episode about it, so stay tuned. Well, if you're here because you want to learn more about rapid diagnostics, this is the episode for you, and we're going to be jumping into it. I just want to say real quick, this episode, it was recorded in ASM Microbe 2025 in Los Angeles, and during this conference, one trending theme was rapid AST and rapid identification systems. So within the realm of rapid diagnostics, this is very important. So this episode is about that, and also about ASM Case Reports, the journal, because Dr. Burnham, who's the guest here on this episode, she is the editor-in-chief. And one thing I've noticed that sometimes in the podcast we mention products or companies, are mentioned, but just so you know, it is not an endorsement. We are just talking about the science. So let's go ahead and listen to the episode. Hello, everyone. This is a very special episode. It's been recorded right here at ASM Microbe 2025 from Los Angeles. So this is something a little bit different because normally in the past, as you know, when I have recorded episodes in Microbe, they're always been by myself talking about my experiences, but this time I have a guest. And, she's been a guest before, did an episode years ago about Carey-Power testing. Then she was a guest also on the Clinical Microbiology Procedures Handbook. So with me today, I have Dr. Carey-Ann Burnham. Dr. Burnham, welcome to Let's Talk Micro.
Thank you so much for having me. I'm really pleased to be here.
What a pleasure having you here, and thank you. I know that we talked a little bit offline, and it was like a last minute thing, but I appreciate your time. Okay, so it's been a while since you've been on the podcast, so maybe if you can tell the audience a little bit about what you do and anything else that you want to share.
Okay, great. Thank you so much. What I do, that's a great question. I'm pretty lucky. I get to do a bunch of different things. So my day job is currently serving as Chief Executive Officer for Pattern Bioscience, which is a small diagnostics company based in Austin, Texas. I also hold a faculty position as a professor of pathology and immunology at Washington University in St. Louis. And then over the last year, I've served as the founding editor-in-chief for the new journal ASM Case Reports.
Perfect, and we'll be talking more about that, and I'll be asking you some questions about that. So first, have you had a good microbe event so far?
It's been a great microbe, a lot of great science, really fun and innovative sessions. And of course, it's always great to catch up with friends and meet some new ones.
It is, and as I always tell the audience, it's you get to see a lot. You have a great time. It's very busy. So I was talking to someone that were going to experience their first time here, and it's like any advice, and I'm like, well, you have to pick and choose, and you will miss some things because there's so much. So that's just the reality of it. But still, like you said, great place to see old friends, connect, make new ones. So one of the things that I've seen in my time here at Microbe, and so there's like trending, let's say, a lot of regarding rapid diagnostic testing. I've seen a lot of posters, comparison, especially when it comes to the ID and susceptibility testing, where you have these platforms where you can get, like susceptibility results, in 6 to 8 hours and things like that. So those of us that work in the lab, we understand the impact of this but for a general, for the audience as a whole, someone coming into the field, or what is the importance of rapid diagnostic testing?
Yeah, I think it's a great question. So, as you know, because we've talked before, but before I joined Pattern, I was the medical director of microbiology at Barnes-Jewish Hospital for about 15 years. And I think it's a really exciting time to be a microbiologist. There's a lot of innovation, a lot of new technologies coming forward that are enabling us to care for our patients in whole new ways. And I think we've seen some excellent examples of where rapid diagnostics have completely changed the way we do micro. And I think for me, one of the poster child, since bacteriology is my favorite, is MALDI-TOF, where we went from taking a day after we had a colony to identify microbes to an hour, a minute, same shift. Really, our workflow was the only thing limiting us. And not only were we able to identify microbes quickly, it turns out we were able to identify them a lot more accurately. So we were able to get fast, actionable results. And we also learned a lot about the biology of organisms that maybe we had kind of disregarded as commensals in the past, for example. And this is an example of a rapid technique that we saw completely displace the way we used to identify organisms in about 8 years, dramatic improvement. And so there were a number of papers showing that getting that rapid identification helps get patients on the right drug more quickly. However, that's the identification piece. There's still the question mark about susceptibility, right? And we know that that's the most important thing. There's a lot of literature showing that when we have delays in getting patients on the right therapy, patient outcomes are worse. And the right therapy, I think, comes in a couple flavors, right? So there's those patients where they might be undertreated and they need something different or broad spectrum. That's the most acute case. But there's also the situation of getting patients on the most appropriate therapy, which might mean more narrow spectrum. And that has a big impact too. Reduced exposure to unnecessary antimicrobials can help reduce drug-associated events, things such as Clostridium difficile, and it also reduces the increasing burden or selective pressure on antimicrobial resistance. So I think there's reasons as microbiologists we're incentivized this. There's reasons that patients are incentivized for rapid diagnostics, and then reasons hospitals are incentivized.
Yeah, definitely. And like you see sometimes, especially in some of these areas like blood cultures, for example, where it's so typical. And I tell this to the audience. You're working on that bench, and it's very common that you're looking at your patient, passes away before you're even done with your testing. It's very critical. And micro, it's about time. I mentioned this over and over again, but these things take. You set up susceptibilities, and maybe if you read around the clock, let's say you say it in the morning, maybe night shift might be able to result that. If not, it's full 24 hours. And if you send it out, it takes longer. So it's definitely very, very important. So what can you tell us about, and I completely agree with that before I jump to the question, like how things are changing, very exciting times. And in the scale of time, I haven't been doing this that long. I mean, I did start in the pre-moldy tough days, and I have seen all these changes. So I can't even imagine like someone has been doing it for a long time when you started doing a lot of the manual testing and where we have gotten today. It's just pretty amazing. So I know that like in this event, you were like showing up a platform. Maybe we can talk about that. As far as I know, like I saw different posters about some evaluations, like urines, respiratory samples, right? So maybe we can start talking about this platform. How does it work and things like that, and we'll go from there.
Sure, absolutely. So I'll start with a bit of background, and I look forward to your questions as we go through this. So what we are working on at Pattern is a device that takes clinical specimens and directly from clinical specimens determines the organisms that are present and their phenotypic susceptibility quickly. And how quickly varies a little bit depending on the specimen type. But the product that we're starting with is our pneumonia product, and it's same shift results under 8 hours. So, when I think about something that what properties it has to have to really move the needle and how microbiology is performed, I think about a few things. One, I think we have to be able to go directly from the specimen because the culture bottleneck adds a lot to the turnaround time. Two, I think it's really important that the test predicts both resistance and susceptibility. So our genotypic tests often give us a resistance gene, which can tell us what drug we can't use, but it might not definitively tell us what it can. And then the third is the ability to resolve polymicrobial specimens. You know this well from your work in the laboratory. Most samples that arrive in the lab every day don't just have one microbe in them. They have a mixture, right? Either because it's the normal flora that's in that body site plus the pathogen, or because multiple pathogens can cause disease. And so this was really what we had in mind. How do we go directly from specimen? How do we produce a phenotypic result? And how do we handle polymicrobial samples? Does that make sense as a background?
Yes, it definitely does.
So the way we operationalize that is we take the sample and we make it into many, many droplets where each droplet either has 1 or 0 bacteria. Then, if you will, each droplet becomes like its own incubation chamber, and that's where we make our measurements. And so we use those droplets to do both identification and susceptibility. The pneumonia product that we're starting with has a small cartridge. It's about the size of a thick sandwich, and it goes into a modular instrument. And in that thick sandwich, there's different reagents, though. So some have media in them that are more for identification, and some have media that are for susceptibility. Then, in addition to the reagents that are in each of the individual zones, we have 2 metabolic indicators that are present in all the zones, and different microbes digest them differently. So what we end up getting is metabolic patterns or phenotypic patterns, if you will, that are very conserved within a species, but are different between species. And so we do pattern matching to get an identification. So similar to MALDI, but instead of looking at peptides, we're looking at metabolic fingerprints. So kind of the assay will ask itself, do I have droplets that have evidence of bacteria? If the answer is yes, then it will say which of these droplets are like one another, and it will cluster the like droplets together because that will tell us how many different things are present, and then we'll identify the different things that are present. In the same cartridge, we'll compare the metabolic pattern of bacteria that have and have not been treated with antibiotics, and we look at the change. And we use that change to determine the categorical interpretation, susceptible, intermediate, or resistant. So then the output is directly from sample, what microbes are there, what's the phenotypic resistance profile.
Perfect. And that is what you said, the droplets with one organism. So that's what like we call the single cell microbiology technology.
Absolutely. But the report out, even though we're measuring individual cells, we're not reporting out the results of individual cells. We're looking at that population. So we have the cluster of things that are alike and making the measurements there. You can imagine it's analogous to the agar plate, right? If you know you have a mixture of E. coli and Klebsiella, you don't identify each one. You visually, as a skilled technologist, look and say, I have these 2 morphotypes and know how many are there. Similar principle.
Okay. And it gives you said an interpretation. So
Correct.
correct. Okay. And as of right now, for respiratory, and then I've seen some evaluations maybe of urine too, like they're trying to.
Yeah, it's a good question. So we currently don't have a product available for sale. We're still in the development phase, and so we're starting with the pneumonia product that's for use on bronchoalveolar lavage and endotracheal aspirate. And for urine, where we're initially setting our sights, is on a screen. And so this is a screen that very quickly tells you, are there bacteria in the sample, yes or no? We kind of think of it as the next generation of urinalysis, and people are kind of anchored on the fact that most urine screens aren't that good. So sometimes when I say there's a urine screen, people seem skeptical. But we've recently done a nice evaluation looking at urines in both humans and then companion animals, pet dogs and cats, showing very high negative predictive value for ruling out bacteriuria. And because in this case, it's really just an absence or presence, it's much faster. It's a couple of hours.
Okay. So without going like into specifics, and as I mentioned before, we're seeing different products out there and things like that. Maybe so. How does this technology differ from other traditional rapid identification diagnostics?
Yeah, it's a great question. And, I'll start by saying I think there's a lot of great diagnostics out there in general, and they're all a little bit different. I think one of the things that makes this test different is that it has both identification and susceptibility in one. It has the ability to handle polymicrobial. So some of the molecular testing can do polymicrobial for identification, but not necessarily for susceptibility. And then one of the big differentiators is the phenotypic susceptibility. So essentially replacing the destination result that a culture provides today, but helping us get there much more quickly.
So as we talked about all the changes that's happened, and by now pretty much a lot of us have gotten used to having moldy stuff and incorporate to the blood flow workflow. We got used to instruments that will ID things like blood cultures and things like that. So we used to getting that ID now. With susceptibilities, maybe some places, might be like, for example, like where I work at, we do direct bloods and things like that. They still take time. So when it comes to this type of technology, let's say, how do you see it fitting into the current hospital workflow? Is it meant to like complement, or I guess more specifically, do we still culture, or how does that work?
Yeah, it's a great question. And I could imagine in your seat in the laboratory, it probably feels like a lot of new technology that's coming is adding to your workload. So, in many cases, you're doing the things you used to do, and then there's something new. It's really our goal that with our system, it will be able to replace a lot of culture. So, for example, with our pneumonia panel, it covers the vast majority of things that can cause pneumonia. But there's a long list of things that rarely causes pneumonia. And so the way we're thinking about it, and of course, this will have to be backed by the clinical trial data when we have them. But if the sample is negative, you will be done. And we know that about 2/3 of respiratory samples are negative. If it's positive, in most cases, you'll also be done if you have an on panel organism with susceptibility. And then if you have a positive test, but there's not an on panel organism, that small percentage would go on to have plating done on it and have conventional identification and susceptibility. So really, the funnel of the old testing gets much smaller than it had been in the past. I'm curious with your direct blood that you're doing, is the direct blood the disc diffusion methodology?
We validated that, and then they also, like our director validated on the Vitek.
Got it. Got it. I'm curious, do you repeat that testing from a colony, or if it's pure from the blood, do you stop there?
We stopped there. So yeah, great question. So the way that the workflow is right now, so we're on the bench, we see the plates, and they label them like direct susceptibility had it done, so you know to look for it. You have your purity plate, so you're like, okay, this method like detected, okay, equalize the ID, looks like it, if not confirmed by MALDI, let's say.
Mhm.
Susceptibility pattern matches, for example, MSSA, so the susceptibility looks like an MSSA, we just let it go if it's pure, yeah.
Okay, that's great. That's great. So, I mean, what do you feel like is your biggest pain point in that workflow or in your blood workflow in general? What do you wish you could change?
I think that like if this, it's just like I don't know, like the blood bench. It's just sometimes it gets. I do like the fact when we had those direct bloods, they do make the workflow way easier because they work very well, and typically everything matches from the resistant markers being detected to the susceptibility pattern matching. And so those are nice because especially like if you have a nice E. coli, for example, lactose fermenter, non-mucoid, and positive, you do that, check, done. And that culture is done. So it does reduce that time. It makes it easier.
That's great.
Yeah. I think that the bloods, in my experience, they're always, right? So it's either a very good day or a very long day because you have two sets, staff Oreos. So you're testing, set up, refer one to the other. But sometimes you get these cultures like they're very messy or it's not growing and you have to do troubleshooting and that can be time consuming. I think for me, maybe, of course, as we start getting this technology and working more with it, because I'm a fan of Molito, the fact that you can get that ID out there, especially if you don't need to do susceptibilities.
Absolutely.
It's like, let's put it on the moldy, or for example, like let's say group B strep or something like that. Done, okay, and you get those out of the way, and then so it makes life easier. Um, as of right now, maybe it's till I get used to it. The idea of you having something that you're putting a result out there, let's say for AST in 6 to 8 hours, and releasing those results out there before, I don't know, maybe you had a chance to correlate on the plate and see that that's what it is out there. Maybe that's what the only thing that's,
It's a big change, right?
yeah. So I'm like, and I was speaking to someone earlier in the conference, like they do something like this, and they're like, no, we just, we set it up, and then 6 to 8 hours. Sometimes, they doctors call me if it's not ready in 8 hours, and I have to tell them, hey, this used to take 3 days before, so it's been 10 hours. Why are you calling me? But they let it go, and then they verify. They play it, and then they look at the organism, and that's pretty much it. They don't do retesting or things like that. So of course, like with every technology, as I said, it's just getting used to it, and um, but the impact, right? There's no denying if this works well, putting that patient on the proper therapy right away, and just has a lot of benefits.
Yes, yes, we're very excited. I mean, I think the benefits for patients will be huge, and I think once labs become accustomed to the new workflow and are convinced by the data, I think it will be good for labs as well. When I think about respiratory cultures, the respiratory bench is often a big pain point. It can be challenging to train people to work the respiratory bench. Those cultures often have a lot of different isolates that are subcultured and can be very time consuming.
I completely agree. That is my least favorite bench.
Oh dear. It's also a lot of people's least favorite specimen to have to see or plate in specimen receiving as well.
Yes. I know I had that experience with a lot of, yeah, that's definitely the feedback. Sometimes, yeah, from the visual looking at it and then plating it, they don't hate, but I do like a good gram stain on a sputum because you get to see so much, and it's the one place, right? So if you see gram negative diploccocci, like I feel very comfortable calling them, you have NPNs and things like that. So it's just it's a nice quality control slide. You get pretty much a little bit of everything in there.
That's a true story, yes.
Yes. So I have no issues with that. I think it's like you said, they can be very time consuming, and if you have a high volume, and then you get into the point that, and if you have your very sick patients where you have this MDR organisms, and then you have to do this further workup and reflex antibiotics. So it's the fact that there's a lot growing on the plate and it's a lot of work. Um, but other than that, yeah, it's just I think my favorite is the wounds. They can get messy too, but I don't know. Maybe I've been doing them a lot longer, and I do them more than respiratory, so I definitely enjoy them more.
That's great.
Um, and I think we touched on that. Anything that you want to add about about the impact of having a same day, ID and AST?
Yeah, I mean, I can hardly contain my excitement about this idea. So, we know that in the hospital setting, most antibiotics are dosed every 8 hours. There's some exceptions to that, but that's pretty typical. And so the way I've really thought about this is if a patient's critically ill, they will of course be started on antibiotics. Then the sample can come down to the lab, we can do the rapid ID AST, and by the time that second dose is delivered, it can be a directed dose that's needed for that patient. I just couldn't be more excited about that. And then knowing through the course of the treatment that the antimicrobial they're on is the antimicrobial that they will need. So even with conventional diagnostics, we know getting patients on the right drug faster leads to better outcomes. I can't wait to be able to actually have the real world clinical data to see how this is going to transform patient care.
Yeah, definitely very exciting. And now that you mentioned it, thinking about it in those terms about the dosing, it brings that reality, right? So it's like the next dose they can be put on the proper therapy, and just for those of you that are listening in and maybe are just getting started in this field, and when I talk about things like like time, right? So a lot of the traditional methods, so you have to play the organism, and then it takes you 18 to 24 hours, and then from there you set up your susceptibilities, and then provided nothing goes wrong, you have another day. If you need to isolate, that's an extra day. So some of this, sometimes it can be 3 days or longer before you actually achieve that final susceptibility result. So it's a lot of time. So thinking about the dosings and 8 hours, that's a lot of time that patient can be on the wrong therapy potentially.
Absolutely. Maybe I could tell you about a case study we had on one of our posters if you're curious. So, this was a patient. He was a 51-year-old man. He was in the intensive care unit at Barnes-Jewish Hospital and had a tracheal aspirate submitted. So the hospital did their standard of care culture like they do, and the culture actually ended up having 3 multi-drug resistant gram negatives in it. It had a Stenotrophomonas, it had an Acinetobacter baumannii complex, and an ESBL-like Klebsiella pneumoniae. In addition, the plate, and if I had a picture of the plate and I showed it to you and I said, "Is this culture going to take a long time to work up?" I can guarantee you would say yes. It took about 4 days by standard of care to get the final susceptibility reported. And so with the pattern system in the preclinical study that we did, we identified all 3 of the microbes, and we actually accurately reported the susceptibility for the 2 on-panel organisms that there are FDA breakpoints to report it. So I was just so excited about this. It was literally 10 times faster to be able to provide that result. And in patients in that setting, they are typically started on vancomycin and cefepime, and that combination would have been totally ineffective for this patient. The vancomycin would be unnecessary. necessary, there's no gram-positives, and the cefepime would have been insufficient to treat those 3 organisms. So that I just feel like that sort of is an exemplar of what you were just saying about the amount of days and how long it can take.
Wow, 4 days. Yeah, no. Sometimes, yeah, those some of those respiratory, you have all these gram negatives growing on McConkey, and then you add the fact that you have some of that, like oropharyngeal flora, for example, on your blood, and they can be very hard to isolate. I think that one thing that helps, of course, it's now that we have MALDI,
Right.
sometimes you see the text, they go in, okay, they see all these morphologies, they spot them, and then, okay, we'll have a stand, let me sub it to confirm. But definitely, yeah, it can get very, very messy. Okay, and of course, I'll ask you about some closing thoughts, but before we switch gears to the ASM case reports, is there anything else that you want to add to this?
I think I'll just add and say it's been really fun to walk the exhibit hall and see all the different types of approaches that folks are taking to rapid susceptibility. I think clinicians and laboratorians alike are really eager for this, and I think having good solutions in that space will be good for patients. So I'm excited to see when we're all back here next year how all of them have advanced.
Yeah, me too, and yeah, definitely a lot of posters, and as I mentioned, yeah, a lot of comparisons, and they're doing comparing rapid ASTs to like traditional ID methods and different platforms, and it's very exciting indeed. One thing probably next year too, as on a side note, one thing that I saw was from this company that they were using AI to like read plates. So that's also potentially something else that we'll start seeing a lot next year
Absolutely.
as AI starts making leaps and bounds and being incorporated into the lab.
Yes, yes, I'm excited about that too. And I mean, I guess we'll see 20 years from now, but I think there'll always be a place for skilled medical technologists. I think this technology and the AI will be aids, and then a human to tie it together will still be critical to the laboratory workflow.
Yeah, no, I completely agree. You still need that human eye and that experience to say, okay, you're looking at images or things are evaluating. It's even like right now. Um, For example, like people are used, and I use for certain things, like AI, like ChatGPT and all the other ones. How can I draft this email better or sound more
Or
professional,
faster.
faster, or very good? By the way, for those of you like sometimes I take pictures of posters, put it there, and it gives me like, can you summarize this for me, please? Just with a picture, and it gives you the nice summary. So it's kind of nice. But you still have to evaluate it and take things from it and say, okay, maybe this looks good, but not this, and so yeah, we'll still be needed. Okay, so let's switch gears a little bit and talk about the ASM case reports, which from I understand that it turned one this year.
Yes, yes. So given that a journal is a journal and not a person, I guess we got a little bit of creative license on how we set the birthday party, but we decided to have the birthday party at ASM because it was at ASM last year that the journal was officially announced and that the editor-in-chief, being myself, was announced. So we started accepting papers last August, and we published our first issue in January of this year.
Okay, yeah. So right, so definitely ASM is very active, and you have a wide variety of journals, a lot of publications from different topics, not only from clinical microbiology, but a lot. So what inspired the launch of this journal?
Yeah, it's a great question. So, as you were noting, ASM Journals is a fabulous suite of journals. As a society journal, they are very highly respected, highly cited. And with this whole portfolio covering everything from basic bacteriology, clinical microbiology, molecular, environmental, there actually wasn't a home for case reports in this portfolio of journals. And I and others think that case reports have a really important function. It can be a way we can learn about emerging diagnostics, emerging microbes, and they're really important for teaching across the continuum. And so we wanted a home for case reports in the ASM portfolio of journals, and that's really how we got started.
Yeah, and definitely great for teaching, I would say, and as you mentioned, the journals, yeah, a lot of great content that I use a lot for this podcast. Like that, I'm always carrying and seeing what I find and that I think is relevant for the lab population,
That's great.
and I go for there. Um, okay, so let's talk about a little bit more about the journal itself, and you mentioned so case reports, and so what type of cases are included in there?
Yeah, it's a great question. So I think of it as being quite inclusive. We are interested in cases about clinical microbiology, so cases that might have more of a laboratory focus or a microbial focus, diagnostics focus. We're interested in cases that have an infectious disease focus, and we are also interested in cases from animal microbiology. So we have some veterinary microbiologists, for example, on the editorial board as well. And I'm really excited for cases that show things like examples of new therapeutics in action or new diagnostics in action, really an illustrative case study showing the impact on the patient. And sometimes it can be quite a while before a bigger case series is available, but these case studies can help us bring that forward. And then when it comes to the microbiology portion, we're really encouraging authors to include things such as images of the plates, images of the gram stains. So not only telling the story, but having something really tangible that folks reading will be able to take back into their practice. So right now we are primarily publishing case reports, but very soon we'll be launching two other types of publications. One is a case series, so it's more than a case, right? A bit more synthesis. Those are written a little bit more like a research article. And then the new feature that's coming soon, we're calling News You Can Use. And so News You Can Use is a combination of a case report along with some teaching materials. So there'll be a facilitator guide that has some discussion questions that can be used in a classroom along with materials to work through them, and then a brief PowerPoint slide set. And so the PowerPoint isn't just supposed to repeat what is in the case series, but you can imagine, depending on the typical type of article, it might have pictures of the organism, it might talk about the life cycle, it might talk about different lab techniques you could use for that disease state. And so we're imagining people studying for their boards will use these materials, people educating medical technologists at the bench, infectious disease fellows, microbiology fellows. So I'm pretty excited about kind of extending the impact of the case report throughout that feature.
Excellent. Speaking of cases, can you maybe think of one or share one that made you say this needs to be published?
Yeah, it's a great question. There've been a couple that have been really interesting recently. There's been a number across the journal, but recent things are always in the front of our mind. One was an example of a patient in Ohio that had leprosy, and that wasn't necessarily front of mind. So there was some delays in getting to the diagnosis. And that case had a nice example of histology and an explanation of the different diagnostic methods. There was another really interesting case where the Carey-Ann test system identified that the patient had Mycobacterium chimera, and that had been missed by conventional methods. So that was a great case both to help explain that organism, but how a new diagnostic method helped arrive at the diagnosis.
So if anyone was actually interested in writing up a case for case reports, what advice will you give them?
Yeah, it's a great question. So I think when you go to write up a case report, you should really start by asking yourself kind of, what's the point? What do I want someone to learn or take away after they read the case report? And really fold your story around that. Then go and take a look at the instructions for authors to make sure you structure the article appropriately. So for case reports, the abstract section is broken into 3 different brief sections. Then there's an introduction, a case description, and a discussion. So kind of have in mind what you're looking at. For the discussion, really think about how to put your case in the context of the literature, and then always include, to the extent you can, display items. So pictures of the microbes, pictures of the gram stain, charts and graphs that help really drive home your point. And I think that the display items really are important. The text is important, but the display items are as well. So keep all that together. And then I really like titles. I think titles are really important. When you think about getting tables of contents or looking through papers, reading the title helps you decide if you're going to read the rest of the paper. So as you're crafting your article, if the title is impactful and the title conveys the important teaching point of your paper, that will sort of be a hook that will grab people to continue reading.
I completely agree with the titles. That's one thing. As I'm sometimes there are those titles that draw us. Like when I'm researching for the podcast, and I did an episode years ago about Wolf Hartimonis.
Yes, one of my favorites.
And then the article, said like the fly who cried wolf.
Yes.
So that's a great play on words, and it just definitely, I saw that and I'm like, I have to read this.
I think that was one of Trish Simner's papers. That was a good one with a great title.
Yeah. So it was great. I ended up doing like 2 episodes with 2 different cases. I think one of them was with Paul Luthi from Maryland, back in the early days.
Great.
Yeah. So this journal it's open access. And what is the frequency of publications? Is it weekly, monthly?
Great question. Right now it's every other month. Given that we're new, that's the frequency. But as papers are accepted and become available, they're published ahead of the issue online as well. So there'll be the regular release every other month, and then in the current articles section on the webpage, you can see what else is new.
Okay, and so definitely there is a lot of educational value in this. So how do you see it serving as a teaching tool, maybe for trainees and early career professionals?
Yeah, I think it's a great question. I think sometimes when you're getting started, there's so much great literature out there, it can be hard to know where to start. And so I think case reports are a nice bite-sized unit. So I think it can be used for studying. I think it can be used just to really learn about the field and learn what new things are coming down the pipeline as you're starting. I'm hopeful that clinical labs, for example, will use case reports in support of continuing education for technologists in the laboratory. So, yeah, I think when done well, they really serve to illustrate a specific content that people can take away. I'm curious, do you use case reports for teaching in your clinical laboratory?
Um, not much.
No.
No, to be honest, yeah. We don't. We do share sometimes if we see something. I mean, we do have like a bug of the week and things like that, and we do make some presentations like that. But not a lot on the case reports. I mean, it's just always now that we have gene sequencing, so we're seeing all these names,
Oh, yes,
and then
yes,
we use
of
like a
course.
bug of the week. This is it, and then like gram stain and biochemicals, and talk a little bit of what kind of sample it was seen.
That makes sense.
Yeah.
Yeah, that sounds terrific. So you're kind of using cases from your own patient population to serve that need.
Yeah, I mean, I definitely see the value, and I don't know if maybe they'll incorporate it one day. I think that sometimes it's such a fast paced environment that everything moves so quick. So, but we don't know what the future brings for us right now. No, any when it comes to like any particular cases or topics that you're hoping to see more in the future, maybe rare organisms, like resistant mechanisms along those lines.
I mean, all of the above. So, as someone who loves antimicrobial resistance, yes, as we see resistance where it shouldn't be, which, we don't like, but it happens, that's great. The novel diagnostics that we talked about earlier in the podcast, I would love to see cases submitted showing the impact of those on patient care and also on the laboratory workflow. I'm very excited that we're accepting cases from our veterinary microbiology colleagues, and I would like to see more of those. I learn a lot from each of them. There's organisms that are not as familiar to me, and the disease presentations are not as familiar. And then with these novel antimicrobials coming to market, I would also like cases showing how the antimicrobial was used. So, in what patient use case was it appropriate? Were there dosing considerations? Did the interdisciplinary team decide to use it and why? I think all of those would be very welcomed in case reports. And then, yes, as novel organisms emerge, either because they're truly novel or because we're better at naming them, to your earlier point, helping the lab know precisely what to do, or at least starting to extend some guidance about what to do when those are encountered.
Yeah, we definitely have seen, yeah, like a lot of, and I saw some posters here in Microbe. I think like the Mayo Clinic, they're presenting on some new species and things like that. So we're definitely seeing so many names. I do remember the early days, right? So there was only so many organisms on your databases for your instruments and
Right.
your ID cards, and then you will end up sending it out or making some sort of determination, like some presumptive ID. But yeah, we're seeing all these things nowadays.
And as the lab, you want to do the right thing. You really want to understand what that organism is, and is there value to the patient in reporting it, or could reporting it actually cause confusion?
Yes. Yeah, exactly. Yeah. So that's one thing I always advise, and when you get things like that and those weird organisms, make sure consult with someone and make sure that before you go putting things out there and reporting things out there. I think sometimes as techs, we're eager to give as much information, but sometimes we have to take that step back and say, okay, let's consult and see what we can do about this first. Um, So before, as far as listeners, and you talked about a little bit about, you gave advice to someone interested in writing up a case. Um, if someone wants to submit a case, or what's the best way to get started?
To get started with the writing process or the submission process?
Submission process,
The
yeah.
submission process, I would go to the Case Reports website, and I think it's a very user-friendly way to submit it. It'll take you through the steps one by one. We'll ask a few questions, and then it's really a matter of uploading the article. To the extent possible, I would say follow the instructions in the instructions to authors. That will make things flow better. But ASM journals are very author friendly, and so there is a policy of format neutral submission. So, for example, if your references weren't formatted quite right out of the gate, that paper won't get rejected. It would still go through the review process. And if you have a case and you're thinking, I like this case, I don't know, I'm not sure what Case Reports want it, always feel free to reach out to me or one of the other editors. We'd be more than happy to give you advice about if that case seems appropriate. And then if you want some advice about kind of what angle you might take or the focus of the case report, we'd be really happy to help with that too.
And I have to say, when you talk about reaching out, people are always available. And when I reach out to anyone connected to ASM or outside, the responses are always very prompt and people are very helpful. So if you have any questions, like Dr. Burnham said, just feel free to reach out.
I agree. The clinical microbiology community is really welcoming and supports each other and wants the best science and wants the best for patient care.
Yes, I was talking about that in the event that I did this morning, when the whole process about the podcast and things like that. And that's one thing that I was, telling the audience. And if you're gonna get involved in science communication, and don't be afraid, reach out, ask questions. People in this community, they're very friendly. They want to help you out. They want to talk to you and share their science and what they're working on. So it's a very positive experience. So we definitely we have spoken about how things are so exciting and all the changes that have happened, and so what excites you the most about where this field is heading?
Yeah, it's a great question. It's hard to pick just one thing that's exciting me the most, but I think if I had to really distill it into a sentence, is that we now have the scientific tools to be able to deliver laboratory testing in a way we probably always wanted to. We're getting close to being able to provide these really fast, actionable results.
Yeah, that's exciting indeed. I mean, I think as microbiologists, and I don't know how other people do, but like if I have these cases and they're either I'm not able to get a result on time, and that stuff kind of stays with me even after I step outside the lab. And I'm okay that you get that feeling when you're done and you put the right result out there and you're done with this culture and you do it in a timely manner. So that's definitely very exciting, that the way that this is heading, that we'll be able to do that. I'm with you there.
I feel like the future is really bright.
Yeah. So before I ask you a question that I like to ask guests, I started doing this a while ago, and it's always exciting to see what answers do they have. But do you have anything else to add about this or anything at all?
I guess I'm curious, what are you most excited about in the future and what you saw at this meeting?
I am, yeah, a great question. I am very excited, and so I will say about the incorporation of AI and see how we're going to incorporate that and how it's going to work. Um, we do a lot of tests sometimes that they can be very intensive, and if you have things that are incorporated, for example, like reading negatives or things like that, where there's no growth, that it reduces that time that we spend on the bench. That would be definitely a huge plus. And also about this, the rapid diagnostics is very exciting, and see that we're able to, potentially, put that patient on the right therapy, save lives, and those are the two things that are really they're exciting me about.
Great.
I always, when it comes with technology and things like that, I always exhort the people to continue, because sometimes with things like Molotov, and for example, and sometimes some techs, might stop doing the whole critical thinking, and they trust blindly that ID and things like that. So, but it's all a process. But I'm very happy about those two things. Okay, so we all have like an organism that it's either has some significance, anything from a smell, morphology, maybe a mechanism. Some people maybe they did a publication about it. So we tend to have that favorite bug. So I ask you, what's your favorite bug?
Yeah, it's a great question. I have a lot of favorites, so it's hard to pick. But if I really did have to pick just one, it would probably be Corynebacterium, which I know is a genus, not a species. But some of the reasons I like Corynebacterium is because as a genus, it's so variable. We have our lipophilic variants, our non-lipophilic variants, and because it's often underestimated or ignored. But we're learning can be very powerful and has very important disease-specific associations.
Wow, that's a great answer. Yeah, I can see that. Yeah, a lot of times it gets that important. Like it gets okay, like skin flora or things like that, and it's a good one. And I like when it comes to Corine, I always tell the techs, especially when you have those white colonies, beware that you make sure that do a gram stain or something like that. Don't think that people tend to think, okay, growing in blood, not a MAC, it's a cocci. So definitely take the time to do your testing and find out. That is a great answer. And I mean, I'm sure you heard mine, but I'll tell you, yeah, for the audience again, Pastorella still is my favorite, so it hasn't changed.
Excellent. Well, it's good to be open to new possibilities, but to also hang on to your old favorites.
Yes, definitely. Okay. Well, um, Dr. Burnham, thank you so much for taking the time. It's been such a pleasure, and I know that you're definitely very busy, and you took some time to be here and do this episode from with me. So, thank you for taking the time to be in Let's Talk Micro.
Thanks for having me. It was really fun.
What a pleasure. So, here from LA ASM Microbe 2025, this is Let's Talk Micro recording live. So, thank you, everyone. Well, there you have it. I hope you enjoyed this episode. As always, such a great conversation with Dr. Burnham. She's been a guest before, so she always joins the podcast to talk about great topics. And there's definitely value on case reports on the journal, and I hope that this can be used as an educational tool. And when it comes to the matter of rapid diagnostics, this is very important. Those of you that work on the bench, that sometimes, how long it can take to get susceptibilities out there, the challenges of getting identifications, and if we can have systems that can provide this in a faster manner, that will be so much greater for the patients. So I hope you enjoyed the episode. And as you know, the Let's Talk Micro podcast is available on all podcast platforms, like Apple, Spotify, Amazon Music, GoodPod. So I invite you to subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. You can find Let's Talk Micro on social media and Instagram, TikTok, YouTube, and Blue Sky Social as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So either via social media or via email, you can reach out with any suggestions, any feedback. They are always welcome and appreciated. And if you are looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book, available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. Great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, my dear audience, is the end of this episode. I hope you enjoy learning about rapid diagnostics when it comes to ID and susceptibilities, and also about the ASM Case Reports journal. As always, I enjoy sharing this information with you. I had such a wonderful time in Microbe. It's always great to meet people, connect, learn more. So, what a wonderful experience. I had a chance to see Kelly Maddock in person, meet her. That's always a plus. So, I am so grateful. Thank you for the support. Stay tuned. Great things coming your way. So, as always, please continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.